record button. Good morning, everyone. Welcome to our final day of the annual meeting of the UK Center for Topological Data Analysis. And our first speaker is Professor Harrington from the University of Oxford, who will talk about algebraic systems biology. Over to you, Heather, please.
Great. Thanks very much for the invitation. It's great to share some of the work we've been doing. So I'd like to jump right in. So what you see on the screen is a watercolor by David Goodsell, and it shows two cells. And so today's talk is really motivated by problems in molecular biology, and in particular, understanding within biology, and there can be mutations with proteins. So here, what you see is two cells, and there are certain molecules, these proteins, and when there are dysfunctions or mutations in them, this can result in disease. And so I'm going to talk about kind of two different frameworks that we've looked at problems in molecular biology. So the first one is something that's called the ERK-MEK pathway. And then the second problem I'm looking at is related to, also related to cancer and angiogenesis. And so, and that's tumor vascular networks. What I want to, obviously, is a TDA talk, so there's going to be topology in both of these. So to jump in, while it is a math talk, I want to show you how biologists abstract what you saw, this watercolor. And these are, each of these nodes are different proteins or molecules. And the one that we're going to be looking at today is this one here that's in purple. And what I want to emphasize is that in mathematical biology, unlike in physics, for example, where there's some laws that we know, we really don't know what the models are and the mechanisms, which is why these are kind of hypotheses of what these possible networks are. And a way to abstract these networks are models. And so the mathematical models that we look at, each of these molecules can be thought as, we can look at each of these molecules and model them as species. And so these species are the variables in our model. And then we have complexes, and complexes where we can look at these as the left and right hand sides of this chemical reaction network. So these molecular interactions. And so we have the left and right hand sides of these reactions. So you have the stoichiometric coefficients here. So it's how many molecules of species would interact to form another set of molecules. And what I want to emphasize is these reactions are dependent on some parameters. And so we use the assumption of mass action kinetics, which is where the reaction rate is proportional. Everything is well mixed and proportional, and so then you can translate this into a system of polynomial dynamical systems. And so here we're just looking at the species concentrations of these molecules and how they change with time. And so this is kind of the first part of the talk, is how to analyze these types of models. And then the second part of the talk is looking at this with taking into account space. So when you have spatial temporal problems. So it's a very simple formulation to then translate this into this system of reactions. So we have, for an example, 4 different reactions, and we can translate this into these 3 ODEs. And so this is, I think, what you cover in like the first day of mathematical biology. So this is very standard for those who are mathematical biologists, but for those that aren't, I think it's quite nice that you can translate it this way. There's also the field of chemical reaction network theory. It's an area of applied maths where instead of working in this in this nonlinear space, you can work in a linear space, in the complex space. And I'm not going to talk about that in this talk today, other than saying that there's a lot of work that exists. If you're interested in it, actually, there's 2 nice reviews. So Jeremy Gunawadena on his website has a very nice introduction. And then Alicia Dickenstein has also written a nice overview and invitation to chemical reaction network theory for algebraic geometers. So now that we have our model, one of the questions is, what can we say, right? So we write models, and often many of them in mathematical biology, because we want to have some biological insight. So what can we say about that? And so some of the standard approaches are to analyze what the steady states are, and these can correspond to different cell decisions. So whether a cell divides or dies or specializes, called differentiation, for example, or moves. And then we often will reduce the model into the simplest form we can to then analyze it. If we have data, we want to estimate the parameters. And if we have many models, we want to perform model selection, which model best describes the data that we have. And so it really depends on the data. And there are lots of challenges. So we can't often measure all of the species. The data can be noisy. These models are often nonlinear, as I've shown you. We don't know the parameter values, and we have a lot of them. Okay, so so that's the kind of the overview, and then now we're going to jump into 2 different problems. So that's an overview of the biology and what problems we have. We looked at 2 very what looks like a very simple problem, but actually in preparing for this talk today, I was going through some old talks. So if you've seen a variation of this talk, it's actually a bit different than, because I went through all the way back to when I started looking at the ERC-MEC problem, and that was in 2013 when I visited the Schwarzman lab at Princeton. And when I gave a talk on it, I called it the mysterious MEC-ERC data, because it turns out that data was noisy. But when I visited the Schwarzman lab, they had measurements of what are MEC and ERC. And these are 2 different proteins here, and they dysfunction in something like 70% of cancers. So understanding why they dysfunction is really important, and a number of different pharmaceutical companies are very interested in this. So the biology here is actually relatively simple because of the way that the Schwarzman lab set up the experiment. So we have 6 species here, 6 variables, and we have 6 reactions. So we have 6 variables, 6 parameters. It looks like it's a very simple system, but actually the analysis of it ends up being a little bit tricky. So we have this enzyme that is activated, and then ERC is the substrate. They add energy, and then they let it all kind of mix, and then they measure how this ERC, this substrate, is modified, which they call S0 is not modified, S1 is singly modified, and S2 is doubly modified. And then they quantify this, and this is a standard thing, which is a gel. And from this, they replicate this many times. So we have 3 measurements of our species, S0, S1, S2, and 7 time points for each of them. And then for the wild type, which is when MEC is activated, it's activated upstream from other molecules, which is difficult to do the normal wild type, but this is what they do in that experiment. And they do this 11 times. Now, looking at this data, as a mathematician, you might say that's really noisy, but biologists were like, this is so clean. So this is new data compared to what I had seen many years ago. And then just translating our reactions into a full system of equations with conservation equations. So we have some conservation of the amount of MEC and ERC, because it's all very well, it's a very nice system. And We also have information because this system's been so well studied. We know exactly how much of each of, well, this is the experimental setup, but we also have some restrictions on these parameters. So these Ks are the parameters, right? And our data space is the number of replicants. The other thing I want to highlight is that working with Martin Wuhr's lab in Princeton, they then mutated some of the MEC. And why we care about that is one of the mutations is involved in cancer, one of the mutations is involved in development, so embryonic development, and another one is involved both in cancer and development. And then you see another loop over here, and this is related to what is called phosphomimic activation, which is normally what experimentalists use when they are, it's normally what experimentalists use when they're doing these experiments. But so instead, so if you are doing some type of test tube experiment, they would use one of these mutants. Okay, so now we have our full model and we want to analyze the parameters. But often this is challenging. And so there's a lot of work in algebraic model reduction, going back. So Boulier used differential algebra and elimination theory, and then Hubert and Laban used scaling invariance and invariant theory to be able to reduce models. Cassian, Pantea, and colleagues use Galois theory to show when you can do this reduction. But I'm going to focus on some of the work by Gutka, Walcher, and colleagues. And in particular, I have a quote from them, which is being able to reduce, do this quasi-steady state reduction, saying that a precise mathematical description analysis of this quasi-steady state was only achieved in the late 1980s, and some aspects are still not completely resolved. And then they go on to say that there's different, it's complicated by the fact that different groups of scientists, including different groups of mathematicians, have different notions of and different approaches to quasi-steady state assumptions and reductions. And so we are going to use their definition, but there are various different ones that exist in the literature. And their definition is a reacting system is in quasi-steady state with respect to certain species if the rates of change of their concentrations are negligibly small compared to the overall rate of the reaction during some relevant time interval. And so we then use their framework to both come up with the reduction and then to show the accuracy. So I want to mention, though, that model reduction has been around since Briggs and Haldane, so it goes way back. Then Siegel and Slemrod in the 80s worked on this, and both of these, kind of the standard way to think about it is some small parameters introduced, either, and it's kind of called in the framework of singular perturbation theory or separation of timescales. Okay, but using Gutka and All's framework, we first eliminate the variables with the conservation relations. Then we use algorithmic approaches to compute the reduced equation. So in particular, we define this quasi-steady state variety. And there, the whole point is that we can, the reduction here relies on a fact that a variety of dimension q can be represented as zero set of n minus q regular functions in a zero-ski open neighborhood of a simple point. And so then we don't have to define, so often the singular perturbation theory requires us to define the slow and fast variables and kind of a priori know a lot more about the system and the defining equations, and we don't have to do that. So in one step, we can include all this information, and we get not only the rational model, which is the model that one would get through singular perturbation theory, but we would also get what we call the linear ERK model. And this can be obtained, which is how in our first in the biological paper, we were able to come up with this model, but it was kind of a bit more ad hoc and it took a couple steps in deriving it, whereas we can get this in one step. So I guess what I want to emphasize is that these algebraic approaches are very well structured at this quasi-steady state and also assessing the accuracy. So is there anything else I wanted to? Okay, and then so for the biology, the final thing to mention here is that the biologists are very interested in this parameter, and there have been many very kind of high impact papers published in the experimental side, determining if, whether this parameter effectively is large or small, and that affects the mechanisms in the ERK-MEK pathway. Okay, so then the goal here, once we have three models now. We have the full model, we have the rational model, we have the linear model, and then the goal is to estimate these parameters. And the idea is identifiability, is we have some point in an image and we want to know where it came from. And so for each model and experimental setup, we have this model prediction map, and that sends the parameters in our model to our sample space. So if we let some data be a point in the space of observable model solutions, we want to attempt to compute the inverse of this map. So then structural identifiability is determining if this map is injective. And so the main thing that we show is that this linear ERK model with the data that we have, and that's very important, I'm going to keep emphasizing, it really depends on the data we have, that we can show it's generically, structurally, globally identifiable, and practically identifiable. Okay, so now I'm going to need to define a few things. So structural identifiability. Okay, so I just showed you what this model prediction map going from parameters to our solutions, but and we can formulate this as questions about fibers of the map, right, that sends these parameters to the solutions. But it turns out that it's difficult to know and to know much about this specific map at these specific time points that we have for our data. And so instead, what we can look at is what we call an abstract model prediction map, which goes from our parameters to the 3D space of analytic functions in one variable in time. And then we have an equivalence relation that the parameters in this abstract model prediction map are equivalent to those in the model prediction map. These two parameters are equivalent if this parameter is equivalent to that in the from the model prediction map. And then using the theory of Ritt and Kolchin from theory of differential ideals and differential algebra, we can translate this abstract model prediction map into one about the rational map, where we're looking at a coefficient map that we obtain from this theory. And the way that we obtain it is we compute characteristic sets, and then we can be able to test the injectivity of that. And then from this, we can then determine whether the model is structurally identifiable. And there's actually 5 or 6 different notions, but 3 that I want to present here is either this map is one-to-one outside of a set of measure 0, or if you're looking just in a small neighborhood, then that's locally identifiable, or it's infinite to 1, then it's generically unidentifiable. Okay, so being able to test all of this and the identifiability problem goes back to Lung and Glad and Olivier, and there's from the characteristic sets that we, oh, sorry, you saw all those announced. From the characteristic sets that we can compute, we get what are called input-output equations. And so we're really looking at this triangular set of equations, and we want to take that, and we get these input-output equations of here the linear model is very straightforward. to write out because it's linear and there are nice formulations. So there's a theorem by Nikki Meshkat, Seth Sullivan, and Marissa Eisenberg, where we can write this out for what are called linear compartment models. And then we can construct the coefficient map, and it's injective. And the whole point is that since this is generically globally identifiable with perfect data, it means we'd be able to recover the parameters in the model. And the other thing I want to emphasize is that there's lots of software. There's actually been updates to the software during the pandemic. So now there's, I mean, we did these calculations before, but now some of this is actually online and available in other systems. So it's much easier to test. And we could test this with the rational and the full model, because writing these input-output equations actually kind of goes off the page once you're in with these nonlinear systems, rather than this is a linear system. And we show, and we can compute that they're all generically globally identifiable. Okay, so generically globally identifiable means that this is for the kind of the abstract model prediction map, right? But we want to know, is the model identifiable for the data that we have? And the data that we have is at these specific time points, and that is actually an open problem. We're able to show that that we can do this for our initial conditions, but for the specific time points, it turns out that that is actually an open problem. Okay, so now we have looked at structural identifiability, which is recovering the parameters, being able to recover the parameters with perfect input-output data. But really what we have is we have noise in our data. So if we let psi be this probability distribution, so this is a multivariate Gaussian with mean that's given by our model prediction. So by in this case, we have for the linear model, we have the solution of this linear ODE system. And the covariance is multiple identity. And we have our data point said not, and this may not, once we have some noise, right, it may not be in the image of our model prediction map. So we want to determine whether we can obtain a unique maximum likelihood estimate of the parameters. So then we can say that a model is practically identifiable if this maximum likelihood estimate can be obtained with a bounded confidence interval or region at some arbitrary significance level with some assumptions on our measurement noise. And so, so then we actually, we look at the three different models. And so for the linear model, we can write out the actual solution to the system because it's linear. And we then can plot the pre-image of the boundary of each of these kind of ellipsoids. And we see that they're all bounded. And this is the same one, but just rotated. And I think that the significance level we use, the level was 0.05. And so that's what we show is that if it's practically identifiable, then we can estimate the parameters and it's not going to be unbounded. Whereas with the rational model and the full model, those aren't bounded. So the two larger models are practically unidentified. Okay, so now that we have our now the rest of this part of the section, we're going to talk about the linear model, and we want to infer the parameters. So for any model, we'd like to infer the parameters and lighten the data. And so in a statistical framework, we use the likelihood, and maximizing this likelihood gives us the value of the parameters. So the parameters here are still theta, and maximize the probability of observing the data x. And this likelihood function scores different parameters according to how likely the data are given those parameters. And we use a Bayesian formulation. And so in the Bayesian framework, it centers around finding what's called the posterior distribution, which is dependent on the likelihood and the prior, and then also the evidence. And this is calculated by integrating the likelihood over the parameter space. But we can actually look at this as with many settings, like the sampling that we're doing. It's sufficient just to write down the posterior up to proportionality, so we don't have to think about this evidence. So we don't have to take that into account. Okay, so now we infer the parameters, right, which is what I've just had here, and we infer these three parameters, and we also infer the standard deviation. We have uniform priors, and then these are the posteriors for each of the three parameters. And this is, each color represents one of the mutants of the mech. And what I didn't mention is Stas's lab is a fruit fly lab, and how the shape of the fruit fly wing changes based on some of these mech mutants as well. And so this is kind of the measure of mutation in the mech mutants. And then we can also quantify we get the posteriors of each of these parameters, which we can plot as some point cloud, a sample of the posterior in 3 dimensions of parameter space. And there are many different ways of looking at comparing distributions. And so in the biological paper that we wrote, we've said, okay, well, you can see that this E203K here, which is the cancer mutation, is the most different kinetically in the model, and it's over here. But in that paper, we also didn't incorporate this SSDD, which is the black curve here, the black posterior here. Wait, is that the SSDD? No, sorry, it's the red posterior here. It's we didn't incorporate the red posterior, sorry. And so that was a question we had, is how does that compare? We wanted to kind of see, well, what does the shape of these distributions look like, and can we use, could we use topological data analysis, for example, not just for this, but maybe for more complex, complicated posteriors. And so we came across Omer Bobrovsky's really nice paper. So with Omer Cheyenne and Jonathan Taylor, and they wrote this topological consistency via kernel density estimation. And so the idea is that we could take the posterior in the form of samples, compute, we estimate it with a KDE, and then we filter. So we would approximate the persistent homology of this by computing persistent homology of the superlevel sets. So okay, so and then we get a barcode, and we can compare these with a bottleneck distance. Okay, so I think if there's anything else. Okay, so that oh good. And so this is to show you. This is just to show you these computing the persistent homology of the superlevel sets. We get these barcodes for each of the mutants, the kinetics of the mutants, and we compare the bottleneck distance, and we compute the bottleneck distance. And what we find, this is H1, but H0 is also fine with this, with the KDE, and there's some things we have to check, but everything works out fine. And what we show is actually that the SSDD, which is important in the biology, that most experimentalists will use this SSDD mutant as a wild type, but kinetically it is very different than the wild type. And so that wasn't shown before. And so that's kind of the nice biological result from comparing the wild type and these different mutants. Okay. And so from this, then we've looked at specifically one pathway. We started to look at how do you connect multiple network pathways. And so this is a couple different theoretical, but also kind of, there's some theoretical work, but then there's also some I guess, experimental work. So what we see is actually many of these pathways, they're not disjoint. So one question we have is how do some of the results we have work if you glue over them with identifiability, for example, or if you split them? And we have some results in that direction. And then the other problem that we worked on was there was one of the pharmaceutical companies had 3 people spend a year generating breast cancer cell lines. They have different mutations, and then simulating them with effectively different drugs. They're ligands, but they know many of the ligands targets, many drugs target certain kind of synthetic ways of thinking of ligands, different doses and measurements. And what happens is when they are analyzing this, which is nice, I'm bringing this up because it's, we're thinking of multi-dimensional data and beyond just looking at a matrix. This is a tensor of order 5, and we're able to preserve some of the structure and then cluster with some algebraic constraints to find which cell lines and ligands kind of keep are most similar. And we want to keep some constraints on that. So we looked at kind of crosstalk, and then the next thing that we focus on is actually going to the next step, which is jumping into space. So in some spatial temporal problems. And so this is a long list of collaborators. So it's many different projects we've been working on, but this is kind of the most active work for the last, I don't know, the ERC problem I've been working on most of the pandemic on and off with my collaborators, but then these have been ongoing as well. So this is all new, I guess, in some sense, that we started talking about angiogenesis with experimentalists in Oxford more than 5 years ago. And so from this problem of angiogenesis, which is the formation of new blood vessels near a tumor, this is data that's generated as 3-dimensional data that's generated of the blood vessel networks as they've grown to a tumor. And I want to emphasize that this is actually a really hard problem. So this was effectively 2 different DPhils. First was doing all the experiments, and then another DPhil was being able to process and segment all of the data. So from starting discussions to actually getting data like this, it took many years. Bernadette can tell you about that. And then something that often in TDA, we will first just, there's some standard complexes and standard filtrations, but actually determining what the appropriate complexes for the application is difficult. And Bernadette came up with a radial filtration, which is you start at the center of the tumor, and you include the network as you're growing out. And this was motivated by various other work. So looking at neuron morphology work by Catherine Hess and colleagues, and also thinking about when Jacek Brodsky and his colleagues looked at the lung and filtering, for COPD. We were trying to think of directionality in this filtration, what would be most meaningful, and we came up with these spherical shells. Well, Bernadette came up with that. So then we compute persistent homology of this, and we get the connected components, loops and voids. And then from that, we extract out topological descriptors. So we want to interpret it. And one of the things we can interpret is the bendiness, so the tortuosity. You can see if you have more vessels that are coming in and out as you're going on these spherical shells, they're more tortuous. The other thing is that in vascular networks, normally we don't have loops because obviously that's not very efficient for blood flow to go in a circle. But in tumor vasculature, this happens. And then the size of voids. So if you have less vasculature, maybe there's more dead cells in the middle, or just the tumor is less vascularized. So less nutrients can get to the tumor. And this may matter, for example, for treatment, because blood vessels need to go, if you, certain treatments like chemotherapy would go through the blood, and then it kills more cells if it's more vascular. So that is the setup. And then we have data from two different groups. So we had data from Roche, who funded Bernadette's DPhil, but we also had data from oncology. And so there are different types of data, which means they have different depths. And one of the types of data is actually a time course. So you could see how the blood vessels are growing over time in a mouse colorectal cancer, the mouse model. And they had control, but then they also had ways that you decrease a drug to decrease and a target to an agent to increase sprouting, so increase vascular growth. And the other data set is snapshot. So we only have one image per mouse, and that is a different mouse model. And you have control or this other drug. And looking at quantifying this with our loop descriptor, we could show that the DC101 decrease of sprouting, you actually see a decrease in the number of loops. Whereas in the other two, the number of loops continues to increase over time, for days. Whereas in voids, we see that by treating this with some drug here, we get the voids are larger, so there's less vasculature there. So so this is nice, and there's many different ways to quantify vasculature with some network statistics, but this was the first that showed loops and voids. Okay, so this was very challenging to get this data, and so another question we had was, can we look at in silico synthetic data? And this is the Anderson-Chaplin model was proposed in 1998. And the idea here is that you have endothelial cells, and then at the edge are these tip cells. These are like the growing vasculature, and they grow towards a tumor based on these tumor angiogenic factors. So you have a TAF gradient and then a fibronectin gradient. And a very simple model. So there's hundreds of models now of angiogenesis, but a very simple model is, let's put in two parameters called haptotaxis and chemotaxis. Haptotaxis is related to the fibronectin, and chemotaxis is related the tumor angiogenic factors. And we can model the way that these tip cells move. And this is a discrete and continuous model. So the discrete is actually the movement of each of these cells on a lattice. And it can branch or die. And then you get the output is this synthetic data, which is, it's one realization of this model. So we have some synthetic data. You could think of it as a binary image. And at first we thought, okay, we'll use cubical complexes. But then John Nardini, who worked with us, we worked with on this, he said, well, actually, I think what we want is what's called a Moore neighborhood. And so we look at the Moore neighborhood around each point. So if there's a blood vessel, a vasculature there, we keep that. And then we look at the Moore neighborhood. And that determines if any of these in the Moore neighborhood are also have vasculature in that cell, then we would, for example, create an edge, or for any three, we would create a triangle. So this is how we construct our simplicial complex. Okay. And then the next question is, well, how do we filter this? And so we looked at many different filtrations of our simplicial complex. And the one I'm going to talk about here is just filtering for left to right. And we also tried filtering right to left, up and down, and then other types of filtrations as well. But kind of trial and error, because of the way that the blood vessels grow towards a tumor from left to right. And then the right to left also helped give some of the additional information. Oh, let me just go back. So we compute persistent homology of this filtration. And then we tried different vectorizations, and we found the persistent images in dimension 0 with 1's weighting was the best way to quantify different model parameters. So just to recall persistence images, instead of having birth and death pairs, you have birth and persistence pairs, and you blur this with a Gaussian, place a grid on a surface, you create a vector, and then you can use different machine learning vectorization methods. So what we did, we looked at an 11 by 11 grid of parameters, so 121 different parameter combinations. We simulated each of these in the model 10 times. So we have 1,210 different simulations. And then from that, every 7, we took the parameters, and for every 7, we took 7 simulations for each of these, and then we were able to cluster this with k-means clustering. And what we did was we have like into each of these groups. So we see that there's a haptotaxis driven group of, and that's how these simulations look, a chemotaxis driven group, that's how these simulations look, mixed chemotaxis, haptotaxis, and somewhere in between. Okay, so I'm getting close to time, I think. So I'm just going to briefly mention that we also did the inverse problem. So we want to reverse engineer the parameters of the model by fitting to observed data. So what we do is we start with this. We start with the data. Suppose we have, instead of simulated data, here we know it's a ground truth. We start with this data and we want to work backwards. And so we've developed a way to do this using Bayesian inference again. And specifically, we propose something called topological approximate Bayesian computation. And so the way that we, instead of computing persistent homology, we compute extended persistence. So everything that we're looking at in the persistence image is finite. And the idea is that we can then let each of these vectors kind of be a distance between these extended persistence. And then we accept or reject if it's within some distance. And so we can start to get a from our prior distribution to our posterior distribution, which is really nice. And so we start with observed and we go, we can then see that posterior looks really nice. And we compare this to existing image statistics, and the topological ABC outperforms the existing statistics in 2 different ways that you can measure it. Okay, so on that note, I'm just going to wrap up, which is that I looked at dynamics of networks with this ERC-MEC and showed how we could use TDA to look at the posterior distributions, and then analyzing spatial temporal data. I did the forward problem. That's why we spent a lot of time looking at that problem, but we're starting to look at the inverse problem. And remember, this is the inverse problem of the model parameters. And so it's this very nice pipeline where, of course, if your model is too complicated and you can't get information about the parameters, you can reduce it. And it's kind of this, this cycle. A couple announcements. We have a DPhil that we're looking to recruit. So it's funded through the Cancer Science Program for next year, 2022. That's with Xin Lu and Helen Byrne. And then we have a number of postdoc positions. So I'll be hiring in my group. And then we have a postdoc to look at COVID data, and then also with the center. And then, of course, everyone put in your calendar ATMCS. And next summer, I'm running a Women in Math program here in Oxford for any starting women graduate students. And so on that, I'd like to thank you for your attention.
Thank you very much, Heather. Let's thank Heather, please. Very good.